Imagine developing a pharmaceutical drug that could potentially save thousands of lives. The formulation is perfect, stability studies look promising, and clinical trials are progressing well. But there is one hidden risk that scientists must always consider: chemicals migrating from packaging materials into the drug product. These compounds are known as extractables and leachables, or E and L. Even trace amounts of these substances can affect drug safety, stability, and regulatory approval. This is why pharmaceutical companies perform detailed E and L studies to detect and identify these potential contaminants. In this presentation, we'll explore 5 key analytical techniques that scientists use to detect extractables and leachables and ensure the safety of pharmaceutical products. First, let's quickly understand the difference between extractables and leachables. Extractables are compounds that can be pulled out from packaging materials under aggressive laboratory conditions, such as high temperatures or strong solvents. These studies help scientists understand the worst-case scenario. Leachables, on the other hand, are compounds that actually migrate into the drug product during normal storage conditions. Regulatory guidelines from organizations like USP and ISO require these studies to ensure that packaging materials do not introduce harmful impurities into medicines. To identify these compounds at extremely low levels, scientists rely on advanced analytical instruments. One of the most powerful tools used in E and L studies is liquid chromatography mass spectrometry, or LC-MS. LC-MS first separates complex mixtures using liquid chromatography and then identifies the compounds using mass spectrometry. This technique is extremely sensitive and can detect semi-volatile and non-volatile organic compounds at very low concentrations. In real E and L studies, LC-MS is commonly used to detect substances such as plasticizers, antioxidants, degradation products, and other polymer additives. Because of its sensitivity and wide detection range, LC-MS is often considered a core technique for extractables analysis. Another essential technique is gas chromatography mass spectrometry, or GCMS. While LC-MS works well for non-volatile compounds, GCMS is ideal for volatile and semi-volatile chemicals. It is commonly used to detect residual solvents, volatile monomers, and small organic contaminants that may originate from packaging materials. One of the major advantages of GCMS is its ability to identify compounds using large spectral libraries, which helps scientists match unknown peaks with known chemical structures. Because of this capability, GCMS plays a crucial role in volatile impurity screening. Not all contaminants are organic molecules. Some impurities are metal elements that can migrate from packaging components or manufacturing equipment. To detect these metals, scientists use inductively coupled plasma mass spectrometry, or ICP-MS. ICP-MS is extremely sensitive and can detect metals such as lead, arsenic, cadmium, and nickel at very low levels. This technique is widely used to comply with elemental impurity regulations, such as ICHQ-3D. In E and L studies, ICP-MS helps ensure that metal contamination from packaging materials remains within safe limits. Another useful technique is Fourier transform infrared spectroscopy, commonly known as FTIR. FTIR works by analyzing how molecules absorb infrared light, which creates a unique molecular fingerprint. In extractables and leachables studies, FTIR is often used for rapid material identification. For example, if an unknown residue or particle is discovered in a drug container, FTIR can quickly determine whether it comes from polymer materials, additives, or contaminants. This makes FTIR very useful for screening and structural confirmation. The final technique is UV visible spectroscopy, or UV-Vis. Although it is simpler than mass spectrometry methods, it is still valuable for routine monitoring. UV-Vis detects compounds that absorb ultraviolet or visible light, such as certain stabilizers and antioxidants used in polymer packaging. Because the method is fast and cost-effective, it is often used during stability studies and routine quality monitoring. Each of these analytical techniques plays a unique role in extractables and leachables analysis. LCMS detects non-volatile organic compounds. GCMS identifies volatile impurities. ICP-MS quantifies elemental contaminants. FTIR helps characterize materials. UV-Vis enables quick monitoring of known compounds. Together, these technologies provide a comprehensive analytical strategy to ensure pharmaceutical packaging does not compromise drug safety. As drug delivery systems become more advanced, the importance of accurate and sensitive E and L testing will continue to grow. Ultimately, these analytical tools help ensure that every medicine reaching patients is safe, stable, and compliant with global regulatory standards.